proteins are made of chains of amino acids, very, very, very long chains of amino acids. So I showed you this graphic of an amino acid. Here is an amino group. This is the carboxylic acid group. This is the side chain. R is always a variable group. R doesn't stand for any element. It means variable. The alpha carbon is in the center, and that's important because if we arrange things in a certain way, we'll have a right-handed version. If we arrange it differently, we'll have a left-handed version. If we take amino acids and put them up next to each other, we can make chains with them by forming a peptide bond. And I wanted to show you, here we'll start over. Notice there is one amino acid. The carboxylic acid group is connected to the amino group and so forth. When we talk about amino acids, like the amino and the acids, actually the important part, it's not. It's the R group that's actually important. But here the carboxylic acid group is next to the amino group and ready to do something called a peptide bond. Notice that when I make my peptide bond, here is H2O. You see that H2O getting ready to fall out of the equation? So that's called hydrolyze. Hydro, water, lyze, to break down. We can either split the water or put it back together again. So there's my amino acids ready to be put together. And if we take that water group out, we end up with a peptide bond. And so this is how we make polypeptides and peptide chains. By the way, peptide is just another word for amino acid. Amino acids are also called peptides. If we put several peptides together, we have a polypeptide. Oligo means small, so oligopeptides are small chains of amino acids. And we'll have hormones that are oligopeptides, like insulin, glucagon, vasopressin. They're all polypeptide hormones. They're not proteins. They're very small chains of amino acids that are chemical signals. But if we take very long chains of peptides, then they will spontaneously form proteins. We can take single amino acids and make neurotransmitters. We're going to take tyrosine and make epinephrine, norepinephrine, and dopamine, and we'll talk more about that during neurotransmission. So here again is the generic structure of all amino acids, and what I want you to know about every amino acid is it has an amino, and it has an acid, and then a side chain, a variable group, and all of those are attached to the alpha carbon. I don't need you to memorize this stuff. We're trying to navigate. I'm trying to show you where these words come from and how they're connected to each other so this information can start to grow in your minds over the course of your careers. So here's glycine, and notice glycine doesn't even have an R group. What's the R group? Here's my carbon making two covalent bonds, and anything that's missing, it's a hydrogen group, right? So that's why they draw glycine this way. The R group is just a hydrogen. Here's L-alanine. The triangle pointing this way means the carbon's facing towards you. And then the dotted line means that hydrogen group is pointing away. And that's where that left-handed version, L-levo, left, comes from L-alanine. We use the left-handed version of amino acids in the body. Your body doesn't really use right-handed versions very much of anything, and that's where that L word comes from. And so here, it's just a methyl group. That's a carbon, and when anything's missing, there must be hydrogens around it. Well, arginine's very complicated R group with this collection of nitrogens around it. Asparagine, somebody collected a lot of asparagus to make asparagine, isolate asparagine, and notice it has a very complicated R group sitting on the end of it. Notice aspartic acid there is the amino acid part of the acid, and then there's the side chain, which is an acid as well, aspartic acid. Cysteine has a sulfur group on it. Sometimes you'll see proteins drawn and you'll see sulfur bridges. We'll hear sulfide bridging, cysteine bridging, and we'll see protein structures stuck together, chained together with sulfur groups. If you make a protein and then you make another protein and you make another protein and another protein and you can stick them all together, your body sticks them all together using sulfide bridges. And so cysteine is obviously involved in that. Glutamic acid, you might have heard of glutamic acid, glutamate. Glutamine. Histidine, we're going to take histidine and make histamine with it. You guys know what histamine is? You know what antihistamines are. Isoleucine, notice that's a very complicated hydrocarbon chain. So maybe that hydrocarbon chain will get stuck in the membrane center to help anchor it. Right? So that's a very fat-soluble side chain. Leucine, another set of carbon chains, or a carbon chain coming off the R group. And so leucine, lysine, methionine, again, a sulfur group with a methyl group out here. Phenylalanine will turn into tyrosine to make all sorts of interesting things. We have to have phenylalanine. It's an essential amino acid. Essential means we can't synthesize it. We can use the phenylalanine to make tyrosine. So phenylalanine is an essential amino acid. If we can't make the tyrosine in it because we get caught up in the ketone group, we could have phenylketonuria because our bodies cannot make tyrosine. That's where those words come from. Here's proline. Notice my R group comes around and is part of the amino group. And so when we put the chains together, anytime there's a proline, it causes a hairpin turn in the peptide chain. And so when we talk about beta pleated sheets, when we take a quick corner, is due to proline. We'll talk about serine proteases. That oxygen right there is very essential in a serine protease, and so notice the difference in the R group. Threonine tryptophan is what we're going to make serotonin with, 5-HT, 5-hydroxytryptamine comes from tryptophan. There's my tyrosine. It was phenylalanine until we stuck that hydroxide group on it. Your liver's always doing this to aromatic rings. It'll see that aromatic ring and it'll want to stick a hydroxide group on it. Valine. I don't expect you to memorize that stuff. I just want you to see that it's the side chains that are important, because once we put the amino acids together, those little R groups are what are the machines that make the machinery, the complexity of the machinery. So what I wanted you to see is that, look, the amino and the acid stay the same. It's the R groups that are all completely different from each other.